So our goal in today's lecture will be to take and understand a more modern view of evolution, specifically what we're calling evolution as genetic change. And so when we started off, we defined evolution very simply in our first set of notes as the change in the genes of a population over time. Darwin had a bit of a different view. He was talking about a change in a species. He was limiting his mechanism to evolution by natural selection. You guys know by now natural selection, basically organisms that have high fitness, the best traits, survive, reproduce, pass those traits on. Organisms with low fitness don't survive, and thus the species becomes better suited to its environment. There are other ways that we're going to discuss in this lecture that evolution can happen separate from natural selection. The first thing, and a common refrain and misunderstanding with evolution, is that it's not taking care of the needs of an organism. And so when we look at evolution, a lot of people say things like, oh, a given trait happens because that's what the organisms need. We need to acknowledge that, for instance, for this beaver, it'd be great to have this chainsaw arm, right? So they need or want that. That's not what they get. Selection is able to work with the traits that an organism has and amplify or reduce those traits. So it can make a trait more or less common in the population. It doesn't provide a trait because an organism would need or benefit from that trait. It's only working with what an organism has. When we're talking about what an organism has, what we're referring to are the genes that an organism has and the variations within those genes. Now, how can an organism have different versions of genes? Remember, we've talked about mutation and sexual reproduction as ways that new versions of traits or new combinations of traits can appear. Variations are all the differences among the individuals. So these could be differences in size, color, things like that. And that's what we're talking about in the modern view. So Darwin never understood genes. We're adding the understanding of genes, not just the physical expression of the traits, but the genetic origin of those traits. So we should acknowledge that in order for natural selection to work on an organism, it has to be working on traits that are inheritable. Remember, something is inheritable if it's genetically controlled and can move between generations. So the genes that control that trait have to be present in sex cells. Mutations that don't happen or aren't present in sex cells, remember, can't make it to the next generation. And so then those versions of those traits can't be influenced by natural selection. So what Darwin had said, and still true today, is that natural selection would say that the differences that help organisms survive are going to become more common. So those are the things that have high fitness, and differences that don't help something to survive are going to become less common. It's those differences in traits that we need to understand are caused by differences in the genes of the organism. So when an organism survives or doesn't survive, it's really based on the genes that that organism contains within their cells. The understanding that selection changes the genes is definitely a newer idea. We need to remember that Darwin and Mendel knew nothing about each other. And that's important because these are really the two main scientists that we talk about in the course of Living Environment. Mendel being the father or the founding person of the idea of genetics, Darwin being the founding person of the concepts about evolution. And so Darwin didn't know that things were inherited. He didn't have an understanding of traits and that we could predict these things with probability. And Mendel didn't have an understanding of evolution. And so what we can do now is we're trying to use knowledge from Mendel and his understanding of heredity to talk about evolution. And so what we're doing is we're talking about how uh, things about the environment have an influence on the genes that are inside an organism. And so the environment causes changes to the genes of a species and then can lead to new traits or new characteristics. And so the more modern understanding, moving past Darwin, is that natural selection affects the genetic variations of a population. So today, when a scientist is trying to study evolution from a modern perspective, the scientists like you saw looking at the rock pocket mice and the Galapagos finches, they are making sure that they are focusing on what we would define as a population. Remember, population is a group of individuals of a species in a single area. And when they talk about a population, they're going to get even more specific and talk about a term that we've seen before called the gene pool. The gene pool is the total amount or the total variety of genes that the members of a population contain. So in the mice, the gene pool had both mice that had black genes and mice that had brown genes. And so there was a different mix of color genes available within that population. Um, Gene pools have or are need to have two or more different forms of a gene for each inheritable trait, or else there's no variation there, and then we're not really interested in that trait. Selection likely can't work on that trait if there is no variation. For instance, all humans are born with two legs, right? So humans can't be selected to have three legs. That trait isn't in our gene pool. And so that's not a trait that natural selection can work on with humans. So if we go back and we define evolution as changes in the frequency or changes in the genes of a species over time, what we can really focus on is the frequency of those genes. Frequency is a percentage. So what percentage of individuals have a certain gene? When you're looking at changes in the frequency, what you're saying is that over time, this population is getting more or fewer mice that have the black colored fur genes, right? And so if you see a change in the number or frequency of mice with those genes, what you're seeing is evolution occurring or natural selection occurring, that for some reason that trait is being favored or disfavored by factors in the environment. So here's just an example of the mice and the gene pool. So if we look at the gene pool in these mice, they have two genes. They can have the black color version of the gene or the brown color version of the gene. We look at the gene pool, that would be over here. That's just all the genes that are available in the population. And so this population looks phenotypically mostly to be black mice. But if you count the genes, they actually have more brown genes present in this population than black. That has to do because the heterozygotes have one of each. And so there are more pure brown mice than pure black fur mice. And so there's actually more brown genes in this population. Now, let's say that these mice are living on the lava and the brown mice are all getting eaten by hawks, right? So every time a brown mouse gets eaten by a hawk, you lose 2 brown genes. And if 2 brown mice get eaten, then we lose 4 brown genes, and 3, we lose 6. And then you could start looking at, is the gene pool changing? Well, if this was the pattern that was being observed, over time you'd be losing out on brown fur color genes. And so the frequency of black fur color genes would be going up. Black fur color genes would be getting more common. And so that would be the start of natural selection or evolution by natural selection within those species of mice. So going back to our genetics unit, you might ask, well, Mr. Shrout, why are there brown mice and black mice? And with the pocket mouse example, if you remember back to the video that we watched, the black fur color trait appeared because of a mutation, right? And so mutations are some change in the sequence of DNA. We've talked about a lot of ways that mutations could happen. There could be carcinogens and mutagens, which would be environmental factors that cause mutations. It could be mistakes during replication. It could be mistakes during translation or transcription. But a mutation is some change in the genetic sequence or in the DNA sequence that could potentially cause a change in the trait. And that's exactly what happened in the mice, right? There was a mutation in the color gene, which caused black mice to appear. That would have been an example with the mice of a mutation that can affect the fitness of that organism. And in the case of the black mice, in environments that had lava, that increased their fitness, right? Because it increased their camouflage. And that mutation was heritable, so it had to have been present in the sex cell so it can pass on to the next generation. The only other easy way, we're going to say easy way, to get variation is to shuffle the genes around. And this happens when new gene combinations are created during reproduction. If you remember when we talked about reproduction and meiosis and fertilization, we talked about gene shuffling, crossing over, independent assortment. And basically in sexual selection, you're mixing a whole bunch of genes together from two different individuals. And that can allow for new versions of traits. A great place that this was shown was the beak sizes. In the video about the Galapagos finches, they were looking at the different sizes of the beaks. And so when a male finch and a female finch make babies, their genes would get mixed together and reshuffled, and some of their chicks are born with unique beak sizes, either bigger than their parents, smaller than their parents, in the middle of their parents' beak sizes. And those new sizes might be beneficial or harmful depending on the environment and how well that beak is suited to that environment. So those are the two main ways that we produce new variations. So they're sources of variation. We need variations, remember, for selection to happen. If there's no differences among individuals, selection can't work. Now we're just taking it back a step and acknowledging that those differences among individuals are caused by differences in gene sequence. So we're just going to continue referring to the mice and the finches here. So I'm going to label these M as mouse and F as finch ahead of time. Now, the different selections on the genes present in the organisms gets us to this idea, is the trait a simple single gene trait like the mice, or is it a complex polygenic trait like the finches? Remember the prefix poly, you should remember, means many, and so polygenic would be many genes. And then single gene, I hope you realize, means single genes. So when we look at traits like the mouse, there's only two versions of that trait, right? You can be brown or you can be black. And so there's just one gene that's causing the difference in those traits. And so mice have to fall into one of two categories. They can be in category 1 or they can be in category 2. That's it. This creates a sort of a situation in selection where selection can favor one trait or the other, right? If selection favors trait 1, then it will increase. And if trait 1 is increasing, then trait 2 will decrease, right? So one goes up, the other goes down. There's only two versions of this trait. So that is a great example. The mice, right? Black fur color, brown fur color. That's a single gene trait. So one of the phenotypes can be favored while the other is disfavored. And so you see an increase in one and a decrease in the other. The other type of trait is more represented by the finches. And in the finches, there's more than one gene that controls beak size. There's multiple genes that control beak size. And so you get a range of phenotypes. So you can have small beaks and large beaks, but you can also have medium beaks, and you can have medium large beaks and medium small beaks. And so there's a big difference in phenotypes, a lot of variety within those finches. Now, this doesn't make a simple up, one goes up and the other goes down like the mice, but we end up talking about the whole curve. This whole, what's called a bell curve, ends up maybe moving around. So we're going to spend a minute discussing the different types of selection that can happen on these polygenic traits, traits that are controlled by more than one gene. Okay, so this is a great image. It's an older image, if you can tell. It's from UConn, University of Connecticut, a long time ago. I'm going to pause for a second and let you examine this, try to figure out what's going on and what the original photographer was trying to show. Okay, and so hopefully what you came up with, or what you realized, is that down here at the bottom are different heights being listed out for these people. And so these people are in rows based on height. Maybe you even then took a step further and realized that the people in white were females, and that the people in black shirts were males. And this is actually, if I remember right, it's a picture from the University of Connecticut Medical School or School of Nursing or something like that. And they lined them up by height. And what a teacher would then love to ask is, are these people exhibiting a pattern? So hopefully you can see that they are showing a pattern. Now, where students sometimes have trouble with this is you think pattern. Pattern could be 1, 2, 1, 2, 1, 2, right? Well, what comes next? Number 1. All right, well, that's a repeating pattern, 1, 2, 1, 2. This isn't repeating because it's not going up, then down, then up, then down. So it's a little bit different, but this fits my definition of a pattern. And so I like to say that patterns are something that allow for prediction. In this case, the shape of this graph, it falls into generally what we would call a bell curve. It looks pretty much like that phenotypic, the polygenic trait distribution graph. Now, how could you make a prediction? I could ask you, hey, I have a person standing behind this door, and I want you to guess what their height is, and I'll bet you $50. If you guess that their height would be less than 5'0" all the way over here, would that have been a good guess? And I hope that you're saying no. And I think you also would say it would be a bad guess to have guessed that their height would be all the way over here at 6'5", right? I think most of you would have guessed, based on this data, that that person standing behind the door that you've never seen before would be somewhere in the middle, right? Because you're saying, gee, most people are somewhere in these middle heights. So if I have to make a guess about the height of some person I've never seen before, I'm going to guess that they're in those middle values. And so this is called a normal curve or a bell curve, and this is where polygenic traits tend to be found making these shapes. Now, having a bell curve for a trait is actually usually pretty beneficial for the population. We need to acknowledge that through the history of Earth, through all time, environments are always changing. They're changing all the time. There's climate change. There's solar cycles. There's sea level changes. There are ice ages. And so past climates are always different than present climates. And that has been a challenge for the living organisms here on Earth. Imagine you're a tree. When you are a seedling, you have a certain environment. Then 150 years later, the environment doesn't have the same rainfall. you hopefully are able to adapt to that changing in climate. Now, the benefit of variations, the advantage of variations, is that they make it more likely that even if you, the 150-year-old tree, is not well suited to your new climate, that there will be other individuals within your population that are suited to that new climate. So you, the tree, you're not well off, maybe you die, but the tree next to you was able to tolerate a little bit less rain, right? And so it continues to live, and that means that the species hasn't died off. So variations or having more variation make it more likely that populations will survive. We need to acknowledge that if a variation is not present, it can't help survival. So it's a big idea in biology that variations are important to a species. Species that lack variations are more likely to go extinct than species that have variations. And so these graphs, we're just going to try to preview a little bit, are trying to show you on the left here, here's some original distribution of traits for a species, the coloration of snuzzles. Snuzzles, some fake animal. And in this graph, they're trying to say that the snuzzles here are best suited to their environment. So over time, if we look, there gets to be more individuals on the left side here, and the average moves to the left. In this case, snuzzles on both sides are eliminated, and so the curve loses snuzzles from both sides, and you get more of a middle value. And in this curve, the extremes are better off, right? And so you get more individuals at the extremes and fewer individuals at the center. And so that's three examples of types of selection. We'll define those now. So looking at those curves, looking at the curve of the people lined up by their height, we need to realize that natural selection, organisms living or dying, is usually based on their phenotype, the physical expression of the trait that organism has. That could be the height, the beak size, the camouflage color, etc. So survival and reproduction of a species are to determine which alleles are inherited, and based on what alleles are inherited, members of a species in the future either have more of that type of allele or less. So evolution is any change in the relative frequencies of the alleles in a population, and we need to remember individuals can't evolve, populations can evolve. Individuals can't evolve, populations can evolve. We mentioned earlier single gene traits. This is the mice all over again. Evolution of single gene traits is that one of the two phenotypes can be more fit, and it will increase in frequency. What does that mean for the other phenotype? It has lower fitness, and so it will decrease in frequency. Okay, so this is a great example of selection happening on a single gene trait with these beetles. I'm going to pause for a second while you try to answer these two questions. Which phenotype has a higher fitness, and which allele frequency will decrease? Okay, so hopefully for high fitness, you said orange has high fitness. You would know that because orange is becoming more populated over time. So by the time you get to the end down here, you've got more orange. Which allele's frequency will decrease? Hopefully you said green. Why is that? Because the birds are favoring the green version of the trait. That's the one that they're eating. And so the amount of green genes in this population is going to decrease over time, while the amount of orange genes in the population will increase over time. Now the polygenic traits, these are going to be more complicated. We're going to have three types to look at: directional, stabilizing, and disruptive selection. And these are when we had, as we discussed quickly before we got here, the bell curves. Okay, this image illustrates a concept that we have termed directional selection. This is a type of selection of a polygenic trait in which the entire curve slides or moves to the left or to the right because there's a higher fitness at one end of the curve. And so in this example down here with the graph, what they're trying to show is that beaks on the yellow part of the curve, that's over here, have a high fitness. Beaks on the blue part of the curve over here have a low fitness. And so if at time 1 the curve had an average rate here at this red mark, what would be happening is the birds with low fitness are going to start to die. Their genes are going to decrease in the population. The birds with bigger beaks here are going to have a higher fitness. And so over time, the average beak size of that population is going to shift from the red average to the blue average. And so that population, after this event of selection, will have a larger average beak size than it had previously. That's called directional selection. So here's two quick examples of directional selection. One from the fossil record. We have great directional selection evidence of horse ancestors. And so horses used to be much smaller, and then the fossils that we find from them over time get bigger and bigger and bigger. And so it seems like taller or larger body size was being selected for, while smaller body size was being selected against. So the whole species of horse over time shifted to become taller. This other one is an actually observed example in Galapagos finches. In 1976, to give you a little bit of background, there was an island in the Galapagos Islands where some scientists, the scientists that you watched in the videos, Peter and Rosemary Grant, had measured the beak size of every finch on the island, 751 total birds. And then what happened after 1976 is there was a drought on the island where it basically didn't rain for 2 years. And what happened during that time is the population on the island went from 751 birds to 90 birds. That means 660 birds died. Now that's a lot, and that was a pretty dramatic event. But what really became interesting is before that drought, the average beak size was around 9.5 millimeters in depth on the island. And after the drought, the average beak size was a little bit over 10, 10.1, 10.2. And so what that drought illustrated was selection, a selecting event in which larger beak size was favored. And so the average of the whole population shifted to become larger. A whole different category of selection is called stabilizing selection. Stabilizing selection is defined as the bell curve becoming more narrow. Basically, there's high fitness in the middle, here in the middle, in the yellow spots, and low fitness out at the extremes in these blue areas. So over here and over here. And what this does is it applies pressure, selecting pressure, which favors the average individuals. So the average trait is the high fitness trait, and being far from the average works against an individual. A great example of stabilizing selection is clutch size. This is the number of eggs that a bird has. And so it turns out when a bird has the average numbers of eggs, here, here, or here, they have higher survival of their young. So the idea being that if they have too few eggs and a couple of chicks die, then they're losing a high percentage of their offspring. But if they have too many eggs, they probably can't feed all those chicks, and so a couple of chicks are likely to die also. And basically, this is like Goldilocks. There's a number of eggs that's just right, And so for a lot of birds, that's the selecting pressure is to favor the right number of eggs, the average number, which in this example is being given as 4 to 5 eggs. Another great example that I don't have a picture for of stabilizing selection is human size at birth. So I think that you're aware that most babies, if they're born at full term, are probably between 6 and 8 pounds. And so that is the norm. You don't hear about a lot of babies being born a lot less than that or a lot more than that. And I think you can real quickly see that if you're born too small, you might be weak, have trouble breathing, feeding, etc. If you're born too big, you probably have problems with delivery. And so being too small or too big as a human baby is a disadvantage. And being in that 6 to 8 pound range is sort of the moderate value. So that's stabilizing selection. Our last type of selection is disruptive. And in disruptive, it's actually the high fitness is at the extreme. So these extreme versions of the trait are likely to increase, while the middle values of the trait are likely to decrease. And this is the easiest example of selection to think about actually producing a new species. And that's because what can eventually happen here is that your curve can actually split into two curves. And so you get finches that are selected for small beaks and finches that are selected for really large beaks, and you lose out on finches or for medium beaks. And so you can end up actually splitting a population into two separate populations, potentially making a new species. So here's an example of that with the finches. We've got 3 different species, 1, 2, 3. When they live on the same island, so this is one island, their beak sizes are very much separated off. There's no finch with beak sizes in the middle, right? That's because this finch would be competing with that finch if their beaks were too similar, or these two finches would be competing if their beaks were too similar. And so they get pushed sort of away from each other. What gets interesting though, if we look at this green one down here, this is the middle finch living on its own island without any other finches. And you can see that its beak size actually, when it's by itself, would overlap with this smaller finch down here. And so that would show that the ideal size when it's on its own is actually kind of the same size as this other species. But when they live together, it has a different average on the island that they share. Here's another example. If we look at that middle finch here, where it lives somewhere that without the big finch, it has a bunch of members with larger beaks. But then when they're on the shared island again up here at the top, once again, the shared beak sizes are selected against. There's extra competition when you share those beaks. And so it separates them off. So this is disruptive in that when they're together, they push each other away. Same idea here with the butterflies, just sort of with an artist picture. The blue butterflies are being selected against, so these guys are all dying. The white and the purple are happy, so they're increasing. And eventually you can end up with a population that has just white and just purple, blue is gone. And now you might say we've got 2 different species, or at least went from a polygenic trait to a single gene trait. Okay, so we have just 2 ideas left, and these are basically scenarios in which evolution could happen without selection. Darwin never thought about anything like this, but we now have an understanding that there are some situations that species might experience genetic changes when there's no difference in fitness. One example of that is something called genetic drift. Genetic drift is random changes in the allele frequencies of a population leading to evolution without any selection. Now, genetic drift is going to be much more common in small populations because it's more likely to have random events influencing the gene pool when you don't have very many genes. It can also happen when a small group of individuals colonize a new habitat. Colonize means to arrive at a new habitat. And so the special cases of those have unique terms. One term is founder's effect. That's when allele frequencies change because of who makes it to a new location. And the other is the idea of a bottleneck, and that's having a small population, and so you lose a lot of genes by sort of random influences. Here's a real quick example of how genetic drift could happen. Imagine you had some population of beetles that only had 9 individuals, and Bob walks through the woods and steps over a log, and he squishes 2 beetles. And you think, hey, no big deal. It was just 2 beetles. What we didn't realize is this is an endangered population, and he actually squished 2 beetles that were both carrying a version of a trait. In this case, a not common trait in that population being green color. So after Bob leaves, we have a very different frequency of the green genes in this population. We lost a lot of green genes when Bob squished those bugs. And so when we move forward, the gene pool doesn't have very many of that gene anymore, and that could push that population into being a brown colored population just by a random event of Bob walking through the woods. Now, the idea of a founder's effect is more reliant on who makes it to a new habitat. So imagine this time, again, we're working with beetles, and we say that this group of beetles and that group of beetles, somehow they're floating on a log in a river, and they end up on a new island. So each island is starting off with just 4 beetles. But if we look, like none of these all black beetles made it to island B, and none of these gray beetles made it to island A. And so if we go through time, the question has to be asked, will the beetles on island A and B end up looking like the beetles from the original population? Good, and so I hope that you answered no, they won't there. If the island A population didn't have any of these gray beetles arrive at it, we would expect the population that results from it wouldn't have any of those gray beetles. Same thing, if none of these solid black beetles ended up on island B, we would expect that the population that forms there wouldn't have any all black beetles in it. So those previous 2 examples were examples of random genetic drift through founder's effect. This is an example of genetic drift through that bottleneck idea. The idea is you had a population of individuals, then for some unexplained reason or reason that's not important to us, the population declines a lot. And you end up with a very limited number of individuals that survive a certain time. Think ice age, environmental disaster, overhunting with a species that almost goes extinct, or pollution like bald eagles with a species that almost goes extinct. Then as that species repopulates, its population goes back up, but really it can only have the genes that were present here at this low population time. And so some examples, we know walruses were hunted almost to extinction. We know elephant seals. We know cheetahs during the last ice age almost went to extinction. And so they lost a lot of genetic variation during that time. And that can increase the rates, we know, of a lot of different genetic disorders and problems like that, and cause evolution without selection. So it wasn't necessarily a selecting event that crashed their populations, but that's having an influence on the genes present in those populations today. Our last concept is just to acknowledge that 2 real heavy math guys, Hardy and Weinberg, got together and wrote some equations to predict the likelihood of evolution happening. And this is a little bit detailed, but what they found was that if you want to avoid or prevent evolution, there's a list of 5 things that you sort of have to have in a population here. So random mating, large populations, no movement, so that's called migration into or out of the populations, no gene changes, and no selection, no favoring one trait over another. in order for something to not evolve. Now, we should acknowledge that in nature, in actual environments, no population meets all five of these things. So either mating isn't random, right? Because we have females and males choosing who they're going to mate with. Populations are not always large. Individuals always do migrate. So they get up and move, seeds float through the water, and they grow somewhere else. That's migration. That happens in nature. We cannot prevent mutations, and all species experience mutations. And there is usually selection. Some trait is generally better than a different version of a trait that's in that population. So if those things don't happen in nature, what Hardy and Weinberg say is, well, then evolution does happen in nature because the frequencies of genes will change unless you can guarantee those five things.